The next two lectures are going to focus on neuroimaging. This is just a brief introduction. We have an entire module dedicated to neuroimaging later on in the course with multiple lectures for each modality. So this is just a very brief introduction to start getting you familiar with the terminology, some of the abbreviations, and you can go into a lot more detail later. So what is neuroimaging? Neuroimaging is using imaging technology to non-invasively take pictures of or measure brain structure and function. There are multiple types of neuroimaging that you can do. Magnetic resonance imaging, or MRI, is the main one that we're going to talk about, but it's not the only one. There's also PET imaging, positron emission tomography, CT imaging, computed tomography, and all of those machines, PET and CT, are of course different from MRI, but the general idea of what the machine looks like is similar, with a person on a bed being put in the scanner. And lastly, EEG and MEG, that's electroencephalography and magnetoencephalography. So these are some of the modalities that we're going to talk about. And within MRI, there are multiple modalities as well within that we're going to talk about here today as well. So the different types of magnetic resonance imaging, or MRI, that we're going to cover today are T1 weighted MRI, T2 weighted MRI, diffusion MRI, functional MRI while performing a task, functional MRI while at rest, not performing a task, also called resting state fMRI, quantitative susceptibility mapping, arterial spin labeling, and magnetic resonance spectroscopy. And for each of these, we'll just briefly cover what the sort of imaging is, what does it look like, and what are some possible applications of this type of imaging. So first, in the scanner, we're still just taking 2D pictures of the brain, but we take 2D pictures, multiple slices, if you can imagine, as this image shows here. Each one of these lines would be an individual slice. And within those, we take a 2D picture. And once we have enough 2D pictures, we can combine them together to reconstruct a 3D volume that we can use for our measurements. The unit of measurement that we talk about a lot in imaging is a voxel. This is very much like a pixel in an image. It's just the individual box size. How good is your resolution? So in this example here, each of these boxes is 3 millimeters in the brain. Often, especially with some of the with the standard anatomical imaging that we get, a pretty standard resolution is 1 millimeter cubed. So brain images can be made up of thousands of voxels or hundreds of thousands of voxels, depending on how small the voxels are. And each voxel may represent 1 million brain cells. So we are very much approximating what the brain is doing. We're not directly measuring individual brain cells here. Starting off with the basic MRI, T1 weighted MRI. In T1 weighted MRI, you know you're looking at a T1 image when the fluid, like the CSF, is dark, the white matter is white, and the gray matter is gray. And this can be used to measure volume and related metrics like cortical thickness, surface area, and is often used as a target, a registration target for other images, so that you can know that you're looking at a particular brain region when you're looking at a different type of imaging. You can bring all of them into the same space so that you have multiple different types of pictures of the brain all together. So one example application here. One of the ways that we can measure the brain is we can extract what's called cortical thickness or cortical surface area and volume. Those are 3 related metrics. And we do this by mapping the borders of the peel surface here in red and the gray matter and white matter boundary here in yellow. From this, we can get a measure of cortical thickness, and we can also extract surface area and volume. And one example application of this is this paper here, where they found that alcohol dependence was associated with widespread decreases in cortical thickness. T2 weighted MRI. Also an anatomical scan, so generally looks kind kind of similar to the T1, except it's inverted. So fluid looks white, white matter looks dark, and gray matter still looks gray. And one thing that T2 weighted MRI is good for is bleeds, as shown here. T2 weighted imaging can detect an active bleed in the brain, which would not necessarily show up so clearly on something like a T1 weighted image. Diffusion MRI. Diffusion MRI is a little bit different from these last two in that it's mapping the movement of water in the brain. And we're taking multiple pictures, but from different angles. When you're looking at a picture, the direction that, if it's dark, that means that there's more movement in that area. And so if you imagine you're trying to assess how quickly cars are moving on a road, just taking a picture at one location is not necessarily going to help you. But if you can have multiple views, you can assess how fast things are moving. And so that's what we do with water, which is constrained generally within the neuron. So that's what we're looking at. Areas that have more movement are like the ventricles, where water is moving freely. That's in CSF, moving freely and not constrained by myelin. And we can use this to assess white matter organization and model the white matter tracts. Diffusion MRI has multiple variations, multiple different names. They're all variations on the same concept, but they do differ in specifically how they work. But you might come across the terms diffusion tensor imaging, diffusion weighted imaging, diffusion MRI. And here, this is what a raw diffusion MRI scan looks like, just for one slice, for one volume. From this, we can extract information about how water is moving in the brain. And that's what we can see here from this image, where the colors correspond to the direction of water movement, the primary direction. So here in the corpus callosum, where things are generally moving left-right, it's red. In the blue tracts, like the corticospinal tract, that's where water is moving superior, inferior, and then green is water that is moving anterior to posterior. So there are multiple measures of white matter organization that can be calculated from diffusion MRI. The one that we use most often is fractional anisotropy, or FA. It's calculated using this equation, which you do not have to memorize. Just know that it is expressing the degree to which water is diffusing along the axon rather than across it. So going back to that image on the last slide where the CSF had a lot of movement, that's an area where we have water that can move relatively freely, and we call this isotropic diffusion, meaning that it's random in all directions and not preferentially along one direction. Whereas in an axon bundle, water is going to have more anisotropic diffusion. It's going to move primarily along one direction rather than all of them equally. We can model this as an ellipsoid in the brain. So when things are moving equally in all directions, it's basically a sphere. But in voxels that are representing a bunch of axon bundles, the water might move more along the length of the axon bundle rather than across it. A few other measures that can be calculated. Mean diffusivity is just the average of diffusion in all three directions. Radial diffusivity is the average of the diffusion in the two non-primary directions, so perpendicular to the axon bundle. And AD, axial diffusivity, is diffusion along the axon bundle, along the primary vector. So when we have this information about the direction and the magnitude of water diffusion, we can use this to connect the dots, essentially. If you have those little ellipsoids at every voxel in the brain, you can line them up to create smooth tracks. And this is called tractography, or diffusion tractography. And this is similar to the image from 2 slides ago that showed the colors. This is showing the same thing, where red fibers are those that are moving left-right, so primarily the corpus callosum. Green are those that move anterior, posterior. So here this is showing the cingulum bundle, and then a lot of the association tracts that run anterior to posterior. And blue are those that move superior to inferior. So generally, the biggest one that we think of is the corticospinal tract, which extends from the cortex, your brain, all the way down the spine out into the periphery. And one example application of diffusion MRI is this study of schizophrenia. All of these red, yellow areas are where the researchers found lower FA, or lower fractional anisotropy in schizophrenia. Along with some of the other measures that they looked at, this suggests that there is myelin loss, and in this case, widespread myelin loss in schizophrenia. Next modality is functional MRI. So instead of looking at structure, now we're looking at function. The appearance is you get, after you've done some processing, you can get images like this, where it will show a cluster of activity that's based on blood flow, a sort of a heat map of where are things active. And what we're measuring here is called the BOLD signal, or the blood oxygen level dependent signal. And that can be modeled here. And this is basically how a region of the brain responds to a task. And it takes a second for the brain to respond. There's a response, and then it resolves. So we say that this is measuring brain function. It's not directly measuring brain function, though. What we're measuring, and what that BOLD signal says, is that we're measuring the ratio of oxygenated to deoxygenated blood. And this is a proxy for brain activity. So when your brain has to look at words, for example, the part of your brain that's responsible for looking at, reading words, and making sense of them is going to be more active. So it's going to require more oxygenated blood. So oxygenated blood flows to that region, and then the ratio of oxygenated blood to deoxygenated blood is tipped one direction. And this is important because oxygen, blood, when it's carrying oxygen, has a different magnetic signal than blood that is deoxygenated. And one example here, in this study of people with post-traumatic stress disorder, or PTSD, they were instructed to downregulate emotions to negative pictures. So in the scanner, you can often show a stimulus to somebody. So in this example, they were showing negative pictures to people to see how their brain reacted to seeing negative things. And they were told to try to regulate their emotions when they saw these negative pictures. While they were doing this, people with PTSD showed increased activity in the amygdala. That's this cluster right here, which is an important region for fear response. So this is relative to people who did not have PTSD. Their amygdala was more active when they were told to regulate their emotions. So this suggests that their emotion regulation systems are impaired, and that might be contributing to, coming from a bit of both, their PTSD. Resting state fMRI. The appearance is similar to fMRI, where you're seeing kind of a heat map of activity, but the focus for resting state fMRI is on correlations in activity rather than the absolute activity. So with fMRI, you're seeing clusters of what's active. Here we're interested in what clusters are active together. This is usually done at rest. So the person is instructed in the scanner to just sit quietly, still, and have their eyes open and just let their mind wander, not think about anything in particular. And what we're looking at is how different parts of the brain correlate in their activity. So your brain is never fully active and never fully at rest. It's always humming, doing all sorts of background processing. And we can look at how certain brain regions hum together. So in this example here, if the region, your kind of starter region, we call this a seed, was the posterior cingulate cortex, and your question would be, where else in the brain does the activity over this 5-minute period, where else in the brain has a similar pattern of activity to the posterior cingulate cortex? And it turns out that one of these regions is the medial prefrontal cortex. So you can see in this graph here, this is showing the activity of these various brain regions over the 5-minute scan. And our seed region, the posterior cingulate cortex, is in yellow. And the other region that we found to be highly correlated with it, the medial prefrontal cortex, in orange. And you can see over the 5-minute scan that these two correlate very well. When they fluctuate up together, and up and down together. Whereas you can look at other regions that are negatively correlated, meaning they're doing the opposite. One of these is the inferior parietal sulcus, which you can see here in blue in the graph. When the PCC and the NPF are down, the IPS is up, and vice versa. And these two are kind of anti-correlated over the course of the scan. So regions that are spatially distant from each other, and in some cases not actually directly structurally connected to each other, can have correlated bold signal over a scan duration. There are numerous networks that have been shown reliably across individuals, so it's not totally random how our brains hum together. We all have very similar networks that have different functions, and you can reconstruct these networks. Even in utero, you can see very early versions of these resting state networks, is what we call. And these are regions that we say that they are functionally connected by looking at their activity. And one example application here is one of the networks that we reconstruct most often is the default mode network, or DMN. And these are regions that are more active at rest than they are during a task. Actually, from the previous slide, the NPF and the PCC are two regions that are typically involved in the default mode network. And in this paper, they found that overactivity of the default mode network, which we see in these kind of yellow-green areas, compared to, we're looking at people who have depression versus people who do not have depression. Overactivity of the default mode network was correlated with rumination. This is one hallmark of depression, where people have a tendency to get stuck in negative thoughts, where they can't pull themselves out of. They just think about what went wrong, why they're feeling so bad, how awful it is that they're still depressed and they can't do anything. And they get stuck in this kind of negative cycle. And it turns out that this is correlated with overactivity in one of these brain networks. We're going to stop there for this half of the lecture, and in the next one, we will continue with a few more modalities. One thing that I neglected to add in the last lecture, which is really important, is that MRI does not involve radiation. PET and CT, which we'll discuss in the next lectures, do involve a small amount of radiation, but there is none in MRI. It's just a really, really big magnet. There's no danger to having an MRI as long as you're properly checked beforehand. Because it's a really large magnet, you cannot bring metal into the room. So, for example, glasses, they can't go into the scan room. And everybody who runs the MRI machines is very well aware of what can and cannot go into the MRI scan room. And we do a detailed check to make sure that people have removed all jewelry, that you aren't wearing any clothes that have metal on them. If you have some sort of an implant in your body due to having had a surgery, usually they use non-ferromagnetic materials. They use materials that will not respond to the magnet. Of course, these are always checked, but usually those are fine. So as long as you have been properly checked for metal, there's no danger to having an MRI. And there are, in fact, some researchers who have had an MRI every day for a few years to look at how things change, and they still only have one head and 10 toes. So it's healthy. It's not dangerous at all.